Okay. All right, so the next section in the outline is some of the commonalities between male and female systems. So I'll go over that next. And initially during development, so this is even before birth, many of the reproductive structures in males and in females come from the same tissue. So basically you start off with like a certain types of tissues, basic tissues, and then they later on in development, they become more specialized. So this refers to the fact that there is like one type of tissue that ultimately will become a certain structure in males. And it will, if the fetus is female, it will become a different structure within females. When you have two things like that that develop from the same type of tissue, they're called homologous. So for example, testes and ovaries are homologous structures. And that just means that they've developed from the exact same tissue initially before birth, but then later they kind of were specialized and became their own structures. Other things that are homologous are the glans penis and the glans clitoris, and the scrotum and labia majora. So those are called homologous structures, which simply means they developed from the exact same tissue. Another thing, and that's what this slide that I'm showing here refers to, are that in both males and females, there are hormones that regulate the production of sex hormones and that regulate the production of the gametes. So, and this is the same in both males and females. And you can see here, the first thing that happens is the hypothalamus. Remember, that is sort of in the middle part of your brain. It releases GnRH, that stands for gonadotropin releasing hormone, and it travels to the anterior pituitary gland and it stimulates it to release two other hormones. So those are the two that are listed next. The anterior pituitary is going to release follicle stimulating hormone, that's FSH, and LH, which is a luteinizing hormone. So these hormones get released and they then travel down to the gonads. So that would be testes or ovaries, depending on male or female. So those same hormones, no matter if you're male or female, both of these get produced, and then they travel down to the gonads and trigger them to release the actual sex hormones. So these are the only hormones that are actually different based on whether you're male or female. So if you're a male and you have testes, then they will secrete testosterone. If you're a female and have ovaries, that will stimulate the release of estrogens and progesterone. But initially, before you get to that last release of hormones, the initial hormones are exactly the same in both males and females. Okay, and then one other thing that is another commonality between males and females is that they both use meiosis, which is a form of cell division to produce the gametes. So you do have a definition for meiosis in your study outline. It is a type of cell division that occurs only in the gonads, and it results in the production of cells that are haploid. So we'll go over the terms diploid and haploid right now. It refers to the number of chromosomes that are in the cells. So if you look at the study outline, the first term is diploid, and that means having the normal number of chromosomes. Okay, the normal number of chromosomes. So it depends on what organism you're looking at, the number of the number of chromosomes that it's going to have. Humans have 46. So if we have cells in our body, and in fact, almost all of our cells are diploid, those cells are going to have the normal number of chromosomes, which is 46. So that normal number consists of two sets of chromosomes. We actually have 23 pairs of chromosomes. So within a pair, there are 2. So we have 23 times 2, which is 46. If a cell is haploid, though, then the cell will have half the normal number of chromosomes. So in humans, the normal number is 23 pairs, which is actually 46. That is a diploid cell. If we have a haploid cell, which has only half that number, then that would have 23 chromosomes total. Okay, so instead of like these haploid cells, instead of having 2 sets of chromosomes, they have only 1 set. And this picture here shows you the phases of meiosis. Basically, what happens is you have a pair of chromosomes here. Here's one, here's the other. They both make a copy of themselves. So now you can see that there are 2 of the blue chromosomes and 2 of the red chromosomes right here in the second picture. So they each make a copy. And then in meiosis 1, that is the first division involved in meiosis. These 2 chromosomes separate. So the blue chromosome goes into one cell, the red chromosome goes into a different cell. And then there is a second division, and that's called meiosis 2. And in that division, the 2 blue chromosomes each separate. Each goes into a different cell. And same thing happens for the red. Each one goes into a different cell. So now what you end up with are 4 cells, each containing 1 single chromosome now instead of 2. You started off with a cell that contained 2 chromosomes, as you can see all the way up at the top. And then what it results in is 4 cells that each have just a single chromosome in them. So this is how gametes are produced. There are no other cells in the body that are produced this way. Every other cell in the body has 46 chromosomes. They are diploid. Gametes are the only cells that are haploid. All right, so that takes care of that section. I am not going to spend time going over male or female reproductive anatomy because it's very simple, straightforward. It's just definitions, and I put all of these definitions in your study outline already. So really no need to waste the time talking about that. So let me go to the next thing I wanted to go over, which is spermatogenesis. I'm just going to talk about the process of sperm production, and then I'll do oogenesis, the process of ova production in females. And then the last thing that I want to do is go over a little bit of the heredity section and talk about Punnett squares. So hang on a second. Let me find, okay. So I'm skipping ahead to the spermatogenesis section. This is maybe like eight pages into your outline. Actually, maybe just 7 pages into your lecture outline. I think it might be on page 7 or 8. Remember, my numbering is a little bit different from yours. So anyway, this is the spermatogenesis section. You guys do have, I think, almost everything filled in for you within this section, but I thought it would be helpful just to go over it because it can be a little bit tricky just because it involves meiosis instead of mitosis, which is the normal type of cell division that you're used to. All right, so remember that spermatogenesis is the production of sperm. And this process begins at puberty, once an individual hits puberty, and it continues throughout the lifetime of the male. So it continues for your entire life, basically, once it begins. So initially what happens is you have cells that are called spermatogonia. And when I have this abbreviation underneath the cell name where it says 2N, or all the way at the bottom it says N, this is the typical way that either diploid or haploid is represented. So 2N means that there are 2 sets of chromosomes, which is normal, and that would be diploid. So these cells are diploid. If I have an N, that means that there's only 1 set of chromosomes, and these cells are haploid. So those are the terms that we just talked about a couple minutes ago. All right, so the cells that you start out with are called spermatogonia. They are diploid. You can see they're 2N. And these are just immature cells that are located within the testes. Specifically, there are like these twisted, kind of coiled up tubes within the testes that are called seminiferous tubules. And these are terms that they're in the study outline. They are defined for you. But this is the location of sperm production. So these spermatogonia are in direct contact with the outer membrane that basically forms the wall of the seminiferous tubules. There are a couple layers of tissue, and they're all the way on the outside, these immature cells. So like I mentioned, they are diploid. They have the normal chromosome number. And basically, just like almost any cell in the body, they will divide through mitosis to make new cells. And they will they continue to do that throughout your life, really up until puberty. So in all the early years, these just divide by mitosis to make new spermatogonia. Then at puberty, spermatogenesis begins. So with spermatogenesis, when the spermatogonia divide, after that point, they produce 2 distinctive types of cells. Instead of producing an identical spermatogonium, they produce either a type A cell or a type B cell. So the type A cells will stay at the basement membrane, and they will just continue to divide and make identical cells, identical to them. So they'll just make more type A cells. These are like the typical spermatogonia that you had originally. So they don't really do anything different. But what is different are the type B cells that are produced. So whenever the spermatogonia divide, they also produce type B cells, and they move towards the center of the seminiferous tubules. And once they get to the middle of that tube, they become what's called a primary spermatocyte. So that is the cell that you're seeing right here, primary spermatocyte. Now, this is when meiosis starts. Sorry, I have a cat jumping on. All right, so this is when meiosis begins. Each primary spermatocyte that you now have in the middle of the tubules, they will go through meiosis. And remember, with meiosis, there are 2 divisions, not just one. So going back to this slide really quickly, with meiosis, you have meiosis 1, where the 2 separate chromosomes go into 2 different cells. And then each copy of the chromosome is then going to move into a separate cell. So those are the 2 divisions you have there. So that primary spermatocyte is going to go through meiosis 1, that first division. So of all the chromosomes that it contains, they're going to separate. Half are going to go into one cell and half are going to go into the other. So now that cuts the chromosome number in half. You go from diploid to haploid cells. Now you have half the number, the normal number of chromosomes within both of these cells. These are now called secondary spermatocytes once you have gone through meiosis 1. Okay, the resulting cells are 2 secondary spermatocytes. And remember that these are haploid. So now these two secondary spermatocytes are going to go through the second division of meiosis called meiosis 2. You can see that right here. And so each one of these spermatocytes is going to divide into two. The resulting cells are called spermatids. So at this point, you have half the normal number of chromosomes in here, but there's two copies of each one. So now the two copies are going to separate. So one copy of each chromosome is going to go into one cell. The other copy is going to go into the other cell. Same thing happens for the other secondary spermatocyte. So in total, you're going to end up with 4 spermatid cells. So we're not quite done yet. This is the end of meiosis and the end of the division. So it results in 4 cells, but these are not actually sperm cells yet. They have to go through a process that's called spermiogenesis, which converts the spermatids into sperm cells. So that basically what's involved in that process is you can see the cell kind of elongates, it stretches out, it forms a tail, which is called a flagellum, and it forms a couple other distinct areas. So this is like the main area that's called the mid piece. The red part at the top is the head. Actually, I'm wrong. The red part at the top is that represents the acrosome. So that's all the way at the top. It's really small and it contains enzymes, which actually will allow, if you have fertilization, they will allow the sperm to penetrate some of the layers that surround the ovum. And then the tail is called a flagellum, and that allows for movement of the sperm. So it moves back and forth to propel it forward. Okay, so that is really all that I wanted to discuss with the male reproductive system. Let me do, because I did spermatogenesis, so let me do oogenesis. That's what occurs in the female. Let me do that quickly, and then I'm going to go to the inheritance heredity section and talk about that really quickly and make sure you know how to set up what's called a Punnett Square. So oogenesis, this is on, I think, page 12 of your outline, 11 or 12. And it's titled, if you're looking in your outline at all, you'll see physiology of the female reproductive system in bold letters. And then right underneath of it is oogenesis. So that is very similar to the process of spermatogenesis that occurs in males, but this is what occurs in females to produce the ova. And it's a little bit more complicated because there are some like pauses along the way. It doesn't just happen straight through like spermatogenesis does. So the definition for oogenesis is the series of events that leads to the production of ova, which are eggs in females. And I mentioned just a minute ago that it's very similar to spermatogenesis, which occurs in males. But there are a couple really big differences between oogenesis and spermatogenesis. So number one, the production of primary oocytes occurs only before birth. So a lot of this process happens before birth. Whereas if you remember with spermatogenesis, it doesn't even start until puberty. So it definitely starts after birth. Another difference is that in primary oocytes, which are these cells right here, meiosis 1 starts, but it pauses and then it doesn't resume. So it's like halfway through dividing and it stops and it doesn't resume doing that until years later. So there's like a huge gap in time there. And then finally in secondary oocytes, Meiosis 2 pauses again. So it starts dividing and then it's going to stop. And it will only finish dividing. It will only be completed if fertilization happens. So, and that actually rarely happens if you think about it. So hardly ever does meiosis 2 completely finish with these oocytes. Okay, so getting back to the whole process of oogenesis, there are a lot of events that actually begin before birth. So initially, the cells that you start out with, remember in males, they were called spermatogonia. So for females, they're called oogonia. And these are immature cells that are in the ovaries within females. They are diploid, which means that they have the normal chromosome number. If you remember, that was the same for the spermatogonia. So these are almost exactly the same sort of cell. And just like with the spermatogonia, these will just constantly reproduce. They'll go through cell division and they'll make exact copies of themselves through mitosis. That's the normal type of cell division. So we have, these will ultimately turn into or divide to turn into oocytes. And just so you can get an idea of what an oocyte is, I'm looking to see if I have a picture. Here we go. So the oocytes are actually the egg cell. And if you look, it's difficult to see, but like right here, if you're looking at the PowerPoint, there is a circle in the middle, like pretty small, and then all these tiny cells surrounding the outside of that little circle. The whole thing is called an oocyte. The actual egg cell, the actual ovum, is just what's like right in the middle. And then surrounding it are all of these tiny like cube shaped cells. The whole entire thing, the egg plus all of those cells, is referred to as a follicle. And in your outline, you have, I think the definitions are provided for you. You have the list of the different stages of follicles, different stages of development, and they're pictured here too. So a primordial follicle is when you have a primary oocyte right in the middle. So that would be like these, or these over here even. And they're surrounded by just a single layer of cells. A primary follicle is almost exactly the same thing. You have the oocyte in the middle, surrounded by a single layer of cells, but now the cells in the primordial follicle, they start out being like really just thin, flat cells. In a primary follicle, they're now cuboidal cells. They are cube-shaped cells, so they're bigger. In a secondary follicle, this is when you have still the primary oocyte right in the middle, but now there are multiple layers of cuboidal cells surrounding it. So that would be like this picture right here. Okay, not just a single layer of cells, but now you can see there are many layers of cells surrounding that primary oocyte. And then a tertiary or Graafian follicle is a secondary follicle that now has developed a fluid-filled cavity. So that would be this here. So you can see the oocyte, you can see the layers of cells all the way on the outside, but now in the middle, there's also a fluid-filled cavity right here. Okay, so now you kind of are familiar with some of the structures that I'll be referring to. When I say oocyte, it's the actual like egg in the middle. And when I say a follicle, it refers to the egg and all of the cells that surround it. Okay, so back to oogenesis. This process begins before birth. It begins in the fetus. And initially, you have just these primordial follicles. So remember, a primordial follicle is a primary oocyte. So the oogonia have divided and they've made primary oocytes. And now these are surrounded by just one layer of flat cells. And there are basically two options for what ends up happening to these primordial follicles. They either undergo apoptosis, which is like a programmed cell death. So it's not that they get hurt or they don't develop properly or something, and they can no longer survive. That's not the case. They're actually totally fine, but it's programmed within their DNA that they're gonna not develop any further. So they just like end up disappearing. And that actually happens to 99% of the primordial follicles that develop within the ovaries. The other option is that they continue to survive and eventually they get ovulated. Obviously, that happens after birth, after puberty. But eventually, some follicles will be ovulated every month with the reproductive cycle. And that only happens to about 1% of the primordial follicles that you initially had as a fetus. Okay, so basically that's where you're at birth. You have primary oocytes surrounded by a layer of cells. They're called primordial follicles, and most of them die. They stop developing and they disappear. So now at birth, a female is born with the supply of oocytes that she will have throughout her lifetime. At birth, you would have about like 2 million maybe oocytes, but by the time a female reaches puberty, she will only have maybe about 400,000 oocytes remaining. So they continue to just disappear. And out of that number, the 400,000, only about 500 of these will eventually be ovulated. So now nothing really changes from birth until puberty, but at puberty, you have the release of hormones, which initiates this process of oogenesis to continue. The primordial follicles will grow and develop into primary follicles. So the oocyte stays the same, but the cells around it get bigger. Eventually, they turn into secondary follicles and then Graafian follicles. Within the primary follicle, the actual egg, the oocyte, will complete meiosis 1. So here is the primary oocyte. It will finish dividing. It will go through meiosis 1, and that produces a secondary oocyte and what's called a polar body. Both of these cells are haploid. So remember, during that first division of mitosis, the number of chromosomes is cut in half. So you go from a diploid cell to having 2 haploid cells now. That's always the case for meiosis 1. You always go from diploid to haploid with that division. So this is different than spermatogenesis, because there, when you had the primary spermatocyte and it divided into 2 identical secondary spermatocytes. Here, though, you don't have 2 identical oocytes. You have a secondary oocyte and you have this tiny little cell that's called a polar body. So with this, it is actually just the result of an unequal division. So during meiosis, the oocytes receive the majority of the cytoplasm, that is like all the stuff that's within the cell. Most of it goes to the oocyte. Instead of being equally divided between the 2 cells, most of it goes to the oocyte. The polar body receives almost none of that. And that's okay, because the polar body eventually is just going to like dissolve and go away. It becomes nothing. This is what's most important. This is what's going to end up forming like the mature ovum, the egg that, I mean, the purpose of this is to have fertilization. So this uneven cell division, giving most of the cytoplasm to the secondary oocyte, is important because it ensures that if the ovum ultimately ends up getting fertilized, it has plenty of nutrients for to last it for the time that it takes for it to travel into the uterus. And that takes at least a week. So it's not going to get any nutrients from the mother until it gets into the uterus and it implants itself and blood vessels grow and you have like the formation of the umbilical cord and everything like that. So in that time in between, you have to ensure that the oocyte, or now it would be called the mature ovum, has plenty of nutrients to survive until that time. So that's why this happens. That's why you have this unequal cell division. Okay, let me hurry up and finish this section because I don't have a ton of time left. I can go until about 9:40. And I want to make sure I have plenty of time to do the heredity section. So one more thing to talk about is ovulation, which is the next thing that would happen. We're right here. We just talked about how meiosis 1 completes. You have a secondary oocyte in a polar body. The secondary oocyte will start to go through meiosis 2, but it pauses. And it pauses until ovulation. So at this point now, we have a Graafian follicle. So it's a secondary oocyte that is within the follicle. And remember, this is now the follicle that has a fluid-filled cavity. It's called a Graafian follicle. And it will, with ovulation, it will rupture to release the secondary oocyte that is inside of it. So that secondary oocyte has started to go through meiosis 2, but it is paused. It's paused partway through. And remember, it will not continue to divide unless fertilization occurs. So if it gets ovulated, it moves into the uterine tube, it goes into the uterus. If there are no sperm present and fertilization can't happen, it is shed along with the endometrium of the uterus during menstruation, and it goes away. If there are sperm present and fertilization does happen, that is the only time when meiosis 2 will continue, and that will result in the formation of an ovum and another polar body. So again, the majority of the cytoplasm is going to go into the ovum and not into the polar body to ensure that this ovum has, like I said, plenty of nutrients for about the week-long time that it takes to implant into the uterus. Okay, so that pretty much takes care of the oogenesis section. So here's another, I have a couple, these are all within the PowerPoint that I've provided you, but here's another slide that kind of shows you a little bit more where the pauses occur. So at birth, this is what you have. You have primary oocytes. And you continue to have these primary oocytes surrounded by those cells until puberty. And then at puberty, these will develop into secondary oocytes. Meiosis 1 completes, you have secondary oocytes. And then only if you have fertilization will you end up getting to this last division where you produce an ovum and another polar body. Okay, so I'm going to stop sharing for a second because we're done with that PowerPoint. And I'm going to open up the heredity one. Get it? Okay. And hold on, I have to share. Okay. So now you should be able to see the development and genetics PowerPoint. I'm gonna skip to the genetics section. Here we go. All right. So let me give you a couple quick little bit of background information, and then I'll get to the Punnett Square thing. So basically genetics, heredity is the study of genes and their functions. When we look at inheritance, we're looking at the passage of traits through genes, which are located on chromosomes, from parent to offspring. We can kind of predict, knowing what we know about chromosomes and genes, if you have two individuals that are having offspring, we can predict what traits, what genes the offspring would have. And we can do that using a Punnett Square. I think I'll have to give you a couple more definitions so you can understand how the Punnett Square works. These are they are defined for you in the outline, but let me go through them really quickly. These are chromosomes. They are made mostly of DNA. It's basically DNA that's wrapped around proteins. And a gene is a segment. A gene is a segment of DNA that has a specific location on a chromosome. So if this whole thing, this blue line, is a chromosome, this is showing you that, there are genes. So there's one here, one here, one here, just at specific locations all along the chromosomes. And because we have two chromosomes, we have a pair for each chromosome. So let me go back here really quickly. We have 23 pairs of chromosomes, remember? So 46 total. And so they're just numbered, chromosome 1 through 23. And we have a pair of each one. So that means there's two chromosomes for each one. So for chromosome number 1, we have two. We have two number 1s. We have two chromosome number 2s. We have two chromosome number 3s. So within each chromosome, so for chromosome, let's just say number 1, you have two different chromosomes. They're both chromosome number 1. One of them came from your mother. It was in the ovum, and the other one was in the sperm. So it came from your father. Okay, so I have maternal and paternal written here. This was the chromosome that came from the ovum. This was the chromosome that came from the sperm. And then when you have fertilization, all the chromosomes come together. So the two number 1 chromosomes go together. The two number 2 chromosomes go together. All right, so there you have genes in the same spot that determine your different traits on each of the chromosomes. And the way that they interact is what determines how it represents itself physically on your body, like whether you have blue eyes or brown eyes, your hair color, your skin color, all kinds of things. Stuff that's happening inside your body, like what proteins you're making, all different things. So another group of terms are homozygous and heterozygous. So, one more thing, actually, sorry to say before I define those. You have, like I was just explaining, you have two copies of every gene, because you have a copy from your mother, like on the maternal chromosome, and you have a copy from your father on the paternal chromosome. So one copy of a gene, so either like this one or this one, is called an allele. So you have two copies of every gene. Another way of saying it is you have two alleles within the gene. So this is an allele for this particular gene, and this is an allele. You have two copies, two alleles for every gene that you have, at least two. Okay, so if you are homozygous, that means that there are two identical alleles for the same gene. They are the same. The alleles are the same. So for this one here, and this is just made up, but for this gene that's right here in the middle of the chromosome, here's an allele for the gene, and here's the other allele. You see how they're identical, right? They're both orange. So this would be homozygous because they're the same. Heterozygous is the opposite. So this is when there are two different alleles for the same gene. So that's the case like here and here. This one's red, this one's white. They're different. So this is heterozygous. Okay. The term genotype refers to all the alleles that an individual has. So if you're asking what someone's genotype is, you would have to say what alleles they have. You could say they have a red allele and a white allele for gene number 1. And for the second gene here, they have an orange allele and a second orange allele. So just describing what alleles they have. The phenotype is the term for the observable physical characteristics that an individual has due to the alleles. Okay, so you might say, if this gene, and I'm again totally making this up, but if this was the gene for hair color, the genotype would be that this person is homozygous and they have two orange alleles for that gene. The phenotype would be the physical characteristics that you see as a result. So the physical characteristic would be that they have orange hair. Okay. So the Punnett square is what we can use to figure out what their potential, what an individual's potential offspring would look like what genes they would have. Typically, and I'm going to go back to this same group of chromosomes, typically when you have two alleles, the most common type of inheritance is that one allele is dominant over the other one. And you would have to be told that, like, you're not expected to just know that, you know, the red allele is dominant over the white allele. So that would be something that you would be told. So here you have, we're just looking at this first gene. You have a red allele, you have a white allele, and the red one is dominant. The other one, when you have one that's dominant, the other one is called recessive. And it is the dominant one that you actually would see. The dominant one completely covers up the recessive allele. So with dominant and recessive inheritance, it's not that you see a combination of the two alleles. One completely takes over the other one. So in this case, the red allele is dominant over the white allele. And this is not true, but let's pretend like this is the gene for, I'll just make it easy. We'll say it's the gene for eye color. So if red is dominant over white, then the individual would have red eyes. Again, we know that's not really true, but just to make it simple, something you can think about and visualize. If red was dominant, it would totally hide the white allele. And so the physical appearance would be red eyes. The genotype refers to the alleles that you have. So there's one red allele and one white allele. They are represented with a letter. And usually you use the letter for the allele that's dominant. So the red one is dominant we use the letter R, and we give the dominant allele is capital, gets a capital letter. The recessive allele gets a lowercase letter. So the genotype would be capital R, lowercase r. That means you have one dominant allele and one recessive allele. Whoops. So the best way, I think, to do this is to kind of like practice. And this shows you the Punnett Square setup. And I'll just explain it on here, and then we can do one or two for practice. So you would be told the genotype, so the genetic makeup, the alleles that the parents have. Okay, so if you have a father that is heterozygous for a particular trait, heterozygous means that the two alleles are different. That means that one is dominant and one is recessive. They're not the same. So you can't have two dominant alleles. You can't have two recessive alleles. You have to have one of each. They have to be different. So there's one, we do one capital P. It's kind of hard to tell the difference between the capital and the lowercase, but this is a capital P and this is a lowercase p. This represents the dominant allele and this represents the recessive allele. So that is the father. He's heterozygous. So that's easy. You know, it's one of each, one dominant, one recessive. The mother, so they're telling you she's also heterozygous. So that's easy. That's exactly the same. One is dominant, one is recessive. Okay, so now you have to set up your Punnett square, and you do a square. So do one big square that has 4 squares within it. And you put, and it doesn't matter if you put the father's genotype on the side or on the top, it's going to come out the same. They put the father on the side. So we have his genotype. We put the first allele beside this set of boxes, and we wrote the second allele beside this set of boxes. So we have the capital P up here, the lowercase p in this row. And then we did the mother's genotype. We put her alleles at the top. So we have the capital P above this column of boxes and the lowercase p above this column. And then what you do is bring the letters down and across. So you bring the capital P down into this box and this box. The lowercase p goes into this one and this one. And then we do the same thing with the dad. So we have the capital P, we bring it into this box and this box. The lowercase p goes into this one and this one. So then at the end, you just look at the result. You have in this square, you have 2 dominant alleles. In this one, you have one dominant, one recessive, one capital P, one lowercase p. In this one, it's this same thing. You have one capital P, one lowercase p. And then here you have 2 recessive alleles, 2 lowercase p's. Let me do just an example with you. So if you have, oh, this is going to be tricky. I'm trying to like use my tools to draw here. All right, so if you have a dad who is, let's see, you have a father who is heterozygous and he has, let's see, he's heterozygous and he has brown eyes. And we'll say that brown is dominant over blue. So there's 2 options, brown and blue. Brown is dominant. So we're going to give it the capital letter P. Blue is recessive. So it gets the lower, oops, it gets the lowercase b. So you would be told that which one is dominant, which one's recessive. So brown is dominant, blue is recessive. And the dad in this case is heterozygous. So think about what his genotype would be. He would be, if he's heterozygous, he has one of each. He has one dominant allele and one recessive allele. So he would be big B, little b. The mother is homozygous recessive. Homozygous means she has the same alleles. Recessive means that she has the recessive allele. And you were already told the recessive allele is the blue allele. So she has two blue alleles. Okay. So here is, this isn't going to be great, but here's the Punnett Square. We're going to put the father's alleles. We'll do his at the top. He was heterozygous, so he's a big B and a little b. We'll put the mother's alleles here. She is homozygous recessive. So that means she has two little b's. And then we have to just bring the letters down and then these across. So the capital B goes down here, down in here. Bring the little b down. Okay, and then we do the same thing for the father's allele. We have this lowercase b. We're going to bring it over into the two boxes that it's next to. Same thing for this one. Bring the little b over here and here. And now we're done. So now we have our potential genotypes for the offspring that they would have. These are the options. So if you're asked, okay, actually, let me stop there. Let's just look at the options. So you have heterozygous. This one's heterozygous. This one's heterozygous. So you have one dominant allele, one recessive allele. And these are homozygous recessive. You have two recessive alleles. So you might be asked, what are the chances that they'll have a heterozygous child? And if that was the question, it'd be a 50% chance, right? Half of them are heterozygous. What if you're asked, what are the chances that they'll have a homozygous dominant child? 0% chance, right? There's no homozygous dominant. What are the chances they'll have a homozygous recessive child?
50%.
Yep, because that's the other two. 2 out of 4 is 50%. Yeah. Or if you just said 2 out of 4, that's fine too. Okay, now you have to think about the phenotype. What would the phenotype be? So this is what they physically look like. And we're talking about eye color in this case. For their offspring, what's the phenotype for this heterozygous?
Brown eyes.
Right. And you know that because you are already told which one was dominant over the other. Brown is dominant over blue. So if there's even one brown allele, that's the one that is expressed, not the blue. So these two children would have brown eyes. And what about the homozygous recessive? What phenotype will they have?
Blue eyes.
Blue eyes, yeah. And really that's the only option. They have two blue alleles. They really couldn't have any other eye color. So they will have blue eyes. So the other thing you could be asked is, what are the chances that they'll have a child that has brown eyes? So we already said half of them are heterozygous. And when they're heterozygous, they have brown eyes. So 50%, there's a 50% chance that their children will have brown eyes, a 50% chance that they'll have blue eyes. Okay. Do you want to I have maybe time to squeeze in one more example. Okay, let's say we have hair color, and I'm just totally making this up because this is crazy, but we'll say that for hair color, the options are yellow and orange. And yellow is dominant over orange. So we're going to pick Y to be our letter because that's the dominant allele. So Y is for yellow. That's a capital Y and a lowercase y. Oops, orange. Okay. The father is homozygous dominant. So what would his genotype be? If he's homozygous dominant.
2 capital Y's.
Yeah, good. 2 dominant alleles, 2 capital Y's. What if the mother is homozygous recessive?
She would have
2 oranges.
Oh, sorry, go ahead.
Yeah, you're right. So two orange alleles. So that would be represented with 2 lowercase y's. Good. And then we just want to see what their offspring are. So we do, and it doesn't matter. You can do mom on top, dad on top. This time we'll do the mother on top. So we'll do the lowercase y. And for the father, we have 2 uppercase. Whoops, uppercase. Okay. So we'll bring these across or bring these down. So we do here, I'll bring these down first. Lowercase y, and then lowercase y. Okay, now we bring the uppercase y across. I always like to do the capital letter first. Sorry, these are not beautiful looking letters, but hard to draw using the cursor. Okay. Okay, so here are potential questions you would be asked. What are the chances that they will have a heterozygous child?
100%.
Yeah, if you look, every single one is heterozygous. Everyone has a capital Y and everyone has a lowercase y. They're all the same. So 100% are heterozygous. So if you're asked, obviously, what are the chances they'll have a homozygous recessive child? 0. Chances of a homozygous dominant child? 0%. What are the chances they will have an orange-haired child?
0% since it's recessive.
Right. Yeah. So the heterozygous genotype is going to give yellow hair because yellow is dominant. So if you have even just one yellow allele, it's going to take over and everything's going to be yellow. The orange is not shown at all. So these all are heterozygous. They will all have yellow hair.